Hi everyone, my name is Kourtney S, and I have the pleasure of being Madison's mentor this magic session. When I first met with Madison, she was really interested in learning about neurodevelopmental disorders because she wants to be a pediatrician in the future. And so what we started with was really just like learning about the tools that are used in science today to study neurodevelopmental disorders, mostly because the tools are really cool and a little bit sci-fi. So it's using pretty exciting to learn about them. And so Madison definitely loved learning about the tools, but was ready to kind of focus in and learn more about a specific disorder. And so she ended up picking autism spectrum disorder, which is a feat in and of itself because it is such a complex disorder, not only in the things that cause it or what's driving the disease, even though there's a lot of it that we still don't understand or know about, but also the patient population just varies so much. And there's just such a range there from things that are felt by the patients or anything like that, how the patient presents. And so really it was great to see her kind of learn about the disorder as a whole and then really kind of focus in on a specific thing. And so that was like the big thing I wanted her to do was to pick a protein or an RNA or something that had been implicated in this disorder and to kind of learn more about it, just to be really true to the scientific process. And so she did such a great job. She went through a bunch of literature, picked out papers herself, and then really kind of like honed in on a specific protein. And so that's what she will present to you today. And I'm really excited for you all to see it. It's been really great working with Madison. She has done such an amazing job, and she has really taken a lot of information in and learned and done a great job at applying her own understanding. So I think today is going to go great. I think Madison's going to do great things in the future. And I'm really excited for you guys to hear her talk today. Thanks.
Okay. Hi, my name is Madison Aldridge, and I'm a 10th grader at the Marymount School of New York. Next slide, please. So my mentor is Kourtney Sprague. Kourtney currently works at the Cincinnati Children's Hospital as a sensory neuron researcher. She's from Dayton, Ohio, which is just outside of Cincinnati, and her hobbies include running, cooking, hiking, and spending time with family and friends. Next slide, please. Going into the MAGIC program, I knew I wanted to learn more about neurodevelopmental disorders. So, for my project, I chose to learn more about interleukin-6, which is a cytokine, and how it impacts a fetus's brain while it's in the womb, which can lead to autism spectrum disorder. Some things I've learned during this project are about human brain organoids, which are collections of cells that are early in organoid development, CRISPR, which is a gene editing tool, and human pluripotent stem cells. Additionally, during my time with Kourtney, we went over reading scientific literature using BioRender, which is a website that allows users to create scientific figures and laboratory techniques. Next slide, please. Some highlights from my time at MAGIC include our weekly meetings, which I always look forward to. During our meetings, Kourtney was always so helpful and open-minded with me, something that made my time extremely enjoyable. Additionally, learning about something that I was passionate about was also an amazing experience. Something that I learned from Magic is that I always need to dig deeper when learning something new. I knew what autism spectrum disorder was, and I knew how it impacted kids. However, I didn't know how kids develop it. So learning about that was something that I was really interested in. One challenge that I faced were that some scientific papers were not available to the public or not published online. So I had to overcome that problem by looking for other papers or looking for other resources. Next slide, please. So now I'm going to be getting into interleukin-6 and its role in autism spectrum disorder. Next slide, please. Interleukin-6 is a cytokine. Cytokines are small, soluble proteins that transduce signals in nearby cells or transmit signals to distant organs. Interleukin-6 has pro- and anti-inflammatory functions, meaning that it can promote or suppress inflammation depending on the situation. Interleukin-6 is involved in a wide range of physiological processes, including organ development, acute phase response, which is the body's initial, rapid, and systematic reaction to injury or infection, inflammation, immune responses, and metabolic regulation. Next slide, please. Interleukin-6 is secreted and synthesized in a number of different cells, including monocytes and T cells, which are both types of white blood cells, fibroblasts, which are the most common cell type represented in connective tissue, and endothelial, which plays a crucial role in maintaining the proper function of the circulatory system. Interleukin-6 signals via a membrane-bound receptors, which are proteins anchored in the cell membrane, and soluble receptors, which are cells that are released into the extracellular space. Next slide, please. Before we get into how interleukin-6 affects the fetus during pregnancy, let's take a look at this diagram of the placenta. In your top left corner is a zoomed-in portion of the ductal layer. The ductal layer is a specialized layer of tissue that develops in the uterus during pregnancy, preparing it for implementation and supporting the developing embryo. It is shed off each month when there is no fertilized egg to support. As we can see, according to the key, there are some T cells within the ductal layer, which is important to note since they do secrete and synthesize interleukin-6. Next slide, please. Interleukin-6 has many different roles in the uterus during different stages in a woman's life. IL-6 is expressed in the human endometrium throughout the menstrual cycle and increases after ovulation and peaks during the mid to late secretory phase. Interleukin-6 production by ductal stromal cells, cytotrophoblast cells, and extravilous trophoblast cells is shown to increase with gestational age, which is the length of pregnancy measured in weeks from the first day of the last menstrual period. Lastly, during IVF, if a woman is undergoing in vitro fertilization, their IL-6 levels will be higher than a woman who became pregnant because of natural conception. Elevated IL-6 levels in a woman who have undergone IVF treatment correlates with implementation success. Next slide, please. IL-6 levels are shown to significantly elevate naturally during the course of pregnancy due to the development of the placenta and the fetus. The concentration of the IL-6 is increased in the amniotic fluid, the cervical vaginal fluid, the fetal membranes, the ducta, and the myometrium and the cervix, which are all circled in red. So as we can see, IL-6 is elevated in various places in the uterus, which all surround the embryo. Now we know that IL-6 is important for function within the embryo within the uterus. However, once these levels elevate past a certain level, they can get dangerous, and this happens due to maternal immune activation. Next slide, please. Maternal immune activation is a state of heightened inflammation in the mother's immune system during pregnancy, often triggered by infections or other stressors. MIA causes additional releases of IL-6, which can be dangerous for the baby's fetal health. The elevated IL-6 levels in a fetal brain disrupts key developmental processes, including laying the foundation for all future brain function, including thinking, learning, and behavior, and disruption can lead to long-lasting or irreversible consequences, such as neurodevelopmental disorders. Next slide, please. Some infections that can cause this increase are influenza virus, which ranges from the common cold to the flu, Zika virus, rubella, a form of measles, listeria, a bacteria, mumps, and fevers. The mother's immune response is passed to the fetus through blood, placenta, and amniotic fluid. Next slide, please. For my demo, I'm going to explain a lab that I went over with Kourtney in a scientific research paper. In this lab, there were mice with increased IL-6 levels that were genetically modified, and then there were control mice with normal IL-6 levels in their brain. The first test that they did on these mice was a neurological reflex test, which included forepaw reaching, writing reflex, trunk curl, whisker twitch, ear twitch, and corneal reflex. The reactivity level of the mice was assessed with tests measuring responsiveness to the petting, the amount of biting, and the vocalization of these mice. This directly relates to ASD because people who have ASD are often more sensitive to stimulus. The second test they did was a cognition test. They used a Morris water maze model to do this. Within a Morris water maze, there is a pool of water, and then there were certain platforms within the maze. They put the mice in the body of water multiple times a day for 6 days consistently. The goal was to see how well the mice remembered where the platforms were. However, the mice with higher IL-6 levels did not remember the platforms as well and were slower to find them. The last test they did was a sociability test. Within the sociability test, there were 3 different chambers which each the mice were placed in for 30 minutes. On one end of the chamber, it was really unfamiliar territory. On this other end of the chamber, there were stranger mice. It was obvious that the mice with elevated IL-6 levels did not want to communicate with the stranger mice as much as the control mice did. One of the key symptoms of ASD is difficulties with social interaction, which is shown through this lab. In conclusion, my time with Kourtney in this magic program has been very fulfilling. I've been exposed to new concepts which have deepened my curiosity about pursuing research in the future. Thank you.
Wonderful. All right, great presentation. Now we'd like to open it up for any questions from our virtual audience. And if you do, anyone joining virtually, you can take yourself off of mute and ask your question to Madison.
Hi, Madison. This is Ira. Wonderful job. Such good research. The question I had is, what was the biggest challenge that you faced during this experiment? I mean, experiments don't often go the way you want to, and that's a good learning moment too. If you could speak to that.
I think probably my biggest challenge during this project was definitely finding the information that I needed to complete this project. A lot of the time, I would click on papers and I would go through it, and it didn't have the information that I wanted. So I would further research and go to other papers on the same website. However, some of them, again, were not publicly available, so I had to overcome that by trying to find different sources or like going on Google Scholar and researching that exact topic or idea.
Thank you.
Great. Well, thank you so much.